coming back to this question, this is your DNA sequence. What's the next line of thinking, Micah? So it's 4 marks. So we would go mRNA, what the effect on the mRNA is, that'd be 1 mark. What the effect on the amino acid is, that would be another mark. And the effect on the polypeptide and finally phenotype would be another mark. Now explain the effect, cause and effect. I guess having the sequence itself will give you another mark. So that's a quick plan that we can have in our answer. So Micah, mRNA, here's your DNA sequence. How do you change DNA to mRNA?
So do you want me to just tell you what it would be?
Yeah, can you tell me, is it going to be identical or complementary? That's what I want to know.
Complementary.
Very
but then
good.
the A, wherever there's an A, that would turn into a U in the mRNA.
Very good. The reason it's complementary is because, Micah, what enzyme reads this DNA to make the mRNA?
RNA polymerase.
Very good. RNA polymerase is going to read this DNA, and the way RNA polymerase works is when it sees a DNA base, it's going to pick up a nucleotide from the nucleus and attach the complementary nucleotide. Okay, so it sees a C, it's going to attach A. Everyone?
G.
Yeah, good. So fill that out for me. You all have 10 seconds. Quickly tell me what the correct sequence and what the abnormal sequence, what RNA will it give you, and how is it going to be different? I'm going to ask one of you to tell me. Let's go as fast as you can without making mistakes. So make sure the big learning point I want you to highlight is it's complementary, the RNA to the DNA. And whenever thymine is supposed to be produced, we don't make thymine, we make uracil. Alec, you mind telling me very quickly what the...
We're just doing the top one, yeah?
Yeah, so that's the normal sequence. What's the mRNA going to be?
G, C, A, U, A, U, A, G, G, A, U, A, C, 4 Gs, A, C, U, G.
Good, done, nailed it. Very good. Next one. Actually, I'll pick someone else. Let me pick Raiden. What did you get for the mutated sequence? Quickly.
GCA, U, A, U, A, G, A, G, A, U, A, C, G, 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 A, C, U, and G.
Very good. Now, one more point. I've basically pretty much told you all the marking plan. 1 mark, student mentions the correct and mutated mRNA compares to 2. 1 mark, student mentions correct and mutated. I'm just going to mute Alec just because of the background noise. 1 mark for student mentions the amino acid sequence, correct and abnormal. 1 mark links it to the polypeptide and phenotype. That's only 3 marks. Where is the last out of the 4 marks going to come from? Let me see who gets this. Yes, Micah?
Identifying that it causes a frame shift mutation.
Very good. You've got 2 different sets of DNA strands. You want to identify what kind of mutation has occurred. It's a point mutation. Do you all agree? You want to identify, is it a point substitution? Is it a point addition or deletion? Very good. Micah would have got 4 marks on that. You see why planning helps? You could have thought you did a really good job just by using this table, but if you don't plan your answer, you will chip away 1 mark every single exam question, which will easily take you out of band 6 range. So point mutation. So what kind, right? So what kind of point mutation occurred? Everyone, what kind of point mutation did occur? For that, you don't even need to look at the mRNA. Look at the DNA itself. Okay, have a system in your head. Maya, how did you come to that answer?
I just read across and then I saw that I was missing one.
Okay, what I would do is I'd first count. What are the total number of letters here? What are the total number of letters here? Maya, what's the count for both of these? I'll get Maya to count the first one. Bilal, you count the second one.
21, I think.
Okay, and for the second? Bilal?
22.
Yeah, the second one has 22. Yeah, good. 21 and 22. So there's been a frame shift insertion mutation here, and specifically a thymine has been added. So you get 1 mark for identifying that thymine has been added. Very good. So we've got the mRNA. Now it's quite easy to read this table. The table is you quite literally get the RNA sequence, GCA, and you go with first base, G, second base C, third base is a A. And you find a table.
Frameshift insertion, is that just another word for addition?
That's it. Yeah, exactly.
All right.
Addition is a
Okay.
frameshift insertion. Yeah. So you can call it
Yeah.
an addition mutation, but you want to identify it's a frameshift mutation. The reason is it's going to change all of the amino acids downstream of that mutation. And you realize that from this question. So yeah, so the first amino acid is going to be alanine. So I'm going to give you all about 30 seconds to do this. Speed completed. Let's go. Okay. Micah, what did you get for the correct amino acid sequence?
So I got Ella,
Do you know what ALA is?
no.
ALA is
Alanine?
alanine.
Yeah.
Yeah. Good. Keep going.
Should I just say the letters? TYR.
Yeah, just say the letters.
ARG.
Tyrosine, arginine. Yep.
ILE.
Yep. So that should be isoleucine. Yep.
ARG.
Arginine,
GLY.
lysine,
And LEU.
leucine. Good. So I'm just saying the words so you start to get a bit more comfortable with what all of these words mean. They stand for one of the 20 amino acids. Good. So that's the normal amino acid sequence. What's the mutated one? Let me get Miriam. Give it a go.
So the first one is still going to be it's going to be ALA.
Yeah.
And then TYR.
Okay.
Third one is going to be ARG.
Look, if you guys want to make your lives easier, it's also fine to just draw lines between every 3 bases. So you can kind of interpret in your head as blocks of 3, right? And that hopefully makes your life a little bit easier.
And then there's ASP.
Good. So ASP is aspartate. Keep going.
THR.
Threonine is the THR. Yep.
GLY.
Glycine.
THR again. And then just a G.
Yeah, and a G will not code for anything because remember, I've been known how many bases code for an amino acid?
3.
Good, right? We only have 1 there, so it's not going to code for anything. So very good. So finally, you need to compare the amino acid sequences. How different are they? How many amino acids are different? You need to analyze this. You can't just get your answer and write it down, right? So how many amino acids are different here?
3.
Good. You need to identify that. A band 5 student would just say, oh, it's quite different, right? A band 6 student would say, yeah, we have 3 different amino acids in these particular locations. And you feel free to circle them if you'd like, right? And so what can you say next? What does this lead to? An abnormal polypeptide. And you want to link it to the final function of the polypeptide. What does it say? Destroys nerve cells in the brain and spinal cord. You get floppy babies. Literally, the babies, they have very poor tone. They can't move around properly. So yeah, you can mention an abnormal protein that causes CNS damage. So now you've linked it to phenotype. Now, if you write an answer like this, you have a bulletproof answer. They cannot take a single mark off you. You've explained what kind of mutation it is, what the mRNA is, what the effect in the amino acid is. You've written the normal and dysfunctional amino acid sequences. You've used the table. You've explained what happens to the protein and finally phenotype. Perfect answer.